Hi guys, it's me. I'm here in Lillard and I'm going to show you how to run a biolog phenotypic array. So what that does is that's going to show us which organism you have down to the species level. So it's not only going to tell you the genus, but it's going to tell you the species. They're a really amazing system. It uses a 96 well plate, and I'll go over that with you. And it basically runs different assays in every well of the plate. And then we end up growing it overnight, and then we can put the results into the computer and it will spit out what our organism is, at least within a certain percentage. So maybe it'll say 85% E. coli or something like that. So we're going to look at two organisms. One is this one here, which is Serratia. We haven't really talked or used this very much, but this is one that we often use in the labs, and students follow this as an unknown. So they figure out that they have Serratia, and we know what that is. So that's a good one to use. And then the other one is my plate. You guys might recognize this. We saw it when I was home, but now it's here. And we're gonna pick one of these colonies, and then we're gonna try to figure out what it is. Okay, so I'm gonna show you how to, first I'm gonna clean the bench space. And then I'm going to take one of these plates. So this is the 96 well plate, and I'll get it open. It already has the things in there at the bottom of each well. So the things meaning mannitol, fructose, glycerol, that sort of thing. And you can see there's an array of 12 by 8. Which is 96. And so there on one side, you have all of the things that potentially an organism might be able to utilize for energy, like the mannitol and the fructose. And then on the other side, you have 3 columns that are things that are bad for the organism, like antibiotics or certain high salt or things like that, things that organisms generally don't want to have. So what we do is, those things are like on the bottom of the well, and we're going to plate a culture of our organism onto each one of these wells. And so that way you'll be able to tell after you incubate it overnight whether it turns purple or not. So if it can utilize mannitol, it will turn purple overnight. If it cannot use mannitol, it will not turn purple overnight. So first we're going to put the Serratia, that's the one we're going to work with first, and we're going to take a swab of it, just as something called an inoculator Z. This is what the company sells, and it kind of just gets you just the right amount of bacteria, and I'll show you that in a second. And then we're going to put it into this IFA media, which that we're going to put into each of the wells. But we're first going to make sure it's about the right concentration of microbe by looking at the absorbance. So first I'm going to take the absorbance of the blank. So this has not had any bacteria put in it, and we're going to see what the absorbance is. So it's way off. It's at 70% transmittance. So we want a blanket at 100% transmittance, which is just about that. I don't know why it's not going all the way, but I think it's okay. Okay, and then we're going to put the organism in here, and we're going to check to see how much the transmittance goes down, right? Because it's not absorbance, it's transmittance. All the light can go through the blank, and then when you add the organism, less light goes through because the organism is blocking the light. Okay, so here's the swab. It's not a usual swab, like not the kind we used in our nose, but it's a special kind of swab and costs a certain amount of money and all that. So that's good. I took a whole colony. I'm gonna swirl it around, and they say to actually wipe it on the bottom because it helps get rid of it, get it off of the swab. So that's what I'm gonna do. And they say to really kind of do it a lot. So that's what we're gonna do. Okay, so now we've put the Serratia marcescens in the media, and I'm gonna like swirl it around. I can even vortex it. And now we're gonna see what our transmittance reads. So we're gonna check the transmittance, and it says it's at 93, and that's good because they tell us if it's between 90 and 95%, then that's the perfect place where you want it to be. So that's great. So now we need to get ready to plate. We're going to use something called a multi-well pipetter. And this is like your normal pipetter, but instead of having just one channel, it has 8 channels. And that's really great because we have 8 channels in here, so we can just more easily add them all and save 8 times the number of pipetting steps. So we'll take one of these troughs. So we took one of these troughs. We're keeping everything sterile. So I'm gonna close this bag. And then we're just going to pour our Serratia in the culture medium into the trough. And then we're going to work with the multi-channel pipetter. But first I want to label my plate. You never want to have a plate that is unlabeled. So I'm going to say Serratia, and I'm going to put the date. Okay. So with the multi-channel pipetter, you just have to be careful that all the tips are on correctly. So I'm putting, this is a little bit tricky. It's really tricky. Just having a little trouble with the multi-channel pipetter there. We got a different one that fits the tips. Okay, so we've got it on, and as you can see, there's 8 tips on here now, but you always want to make sure they're really on firmly. So without touching the very tip, because that'll contaminate, you've got to go and just make sure that they're all on tight, and they are. Okay, so then I'm going to turn this because it's easier this way, and I'm going to put it in to the liquid and draw it up, and then put it into the well. Now, I've done the first column, okay? So I have 11 columns to go. I don't need to change these tips. You could, but I don't need to because I didn't touch the bottom that has any of the solution, like the mannitol or the fructose. I didn't touch that. I didn't get anywhere near it. So I'm going to do this all with the same tips. And I'm going to hold this onto this area here. This has been ethanoled, and it's okay to put it down like this. All right, so that's a little bit, there's some bubbles in there, so I'm going to pick that up again. Some bubbles again, but it's okay. So I'm zeroing it. This is with a different tube. This is the one I just put in. I'm zeroing it to 100. And now I'm taking the bacteria that I just put in. Maybe we'll give it a quick vortex. And we're gonna read the transmittance now. Okay, it's just right at about 95%. So we could add a little bit more, but what this tells me is it's at the high end of transmittance, which means it's at the low end of the cells, but it still is within the okay range. So I'm gonna go ahead and use this, but I'm gonna keep in mind that it may mean I have to stay a little later tomorrow because it might take a little longer for them to grow. And then I'm just gonna pour it in here. And gonna put our tips on. And we gotta make sure, see how that one fell off? Not good. So this is gonna go like this. Not gonna do that, she says. Like this. There you go. There it is. Okay. Again, we have to really push them hard. They do fall off. Like they don't fall off once you push them on, but sometimes you think it's on and it really isn't. Okay. Beth, do they make a 12-channel pipetter?
Mm-hmm.
That would be better.
They're more expensive and harder to use too.
Oh, yeah. That's true. Harder to use. Oops. I missed one. I was talking to you and not paying attention. It's a good idea to look underneath, at the sides so that you don't miss one. All right. Go. All right, and I'm just gonna check to make sure all the wells are even, and I can see that they are, just because I skipped one well and that made me nervous, and so I thought I'd better check, and they are indeed.
Did that get labeled?
Yes, it did. Okay, I'm gonna label this one Kirk's nose. And I'm going to put the date. Okay. And this will go also at 33 degrees. That's the same temperature for all of these types of arrays. And we'll check it tomorrow. So this is going in at 33 degrees. And we're going to put it maybe on this shelf. And say goodnight. All right, it's been one day, and I'm gonna take the plate out and we'll go look and see how it looks. Okay, here we are after 24 hours, and there's our plate. These are the results, and this is all of the reagents that are in each of the wells. So for instance, there's dextrin in A2. So in that assay, you're looking to see whether the organism can utilize dextrin as a carbon source. So all of these things are different things that the organism can grow in, or in the gray areas on the right, there are things that the organism normally wouldn't want to grow in, but maybe these organisms, some organisms are special and can grow in some of those. So let's go and take a look at how to read this. Okay, so there are two important wells in this plate. So there's A1, which is this one here. And A1 is a negative control. So everything that's purple can be positive. So if, for instance, this one is purple, it's positive. This one is a little bit purple. It's more purple than the negative control. So I'm going to say that's borderline. There's three things you can give an organism. It can be positive, it can be borderline, or it can be negative, like this one. B1 is negative to me. So you have to go through and decide what you think all of these different different wells are telling you. Whether it's really purple, that's obvious. Whether it's really negative, that's obvious. Sometimes it's kind of not so strong. So another one I might say is borderline would be like this one here. That one right there is kind of a little bit purple. So I might give that a borderline. Whereas the one next to it is negative. And all of the rest in that line, in that row, are negative. All right, now there's another well besides the negative control that's really important. And this one is A10, which is right here. And this one is positive control. So as you can see, it's really purple, and it always should be purple. And you're assaying for whether the well is about 1/2 to 100% as purple. So for instance, A11 here, I would say it's a little less purple, but definitely is at least 1/2 as purple. Whereas this one is not purple. So this one is not sensitive to the compound that's in here, which happens to be, well, it happens to be growing at pH 6. So this one does grow pH 6, but this one is pH 5 and it's not growing. Some organisms can grow in pH 5, but this one cannot. And likewise, you just go along and assess whether it's 50% as purple, and that means it's not sensitive to whatever's in the well, or it's negative. In this case, a lot of these are negative. Okay, now I'm going to ask you guys to do something, and that is to tell me what you think 2 of the wells are going to look like. And how do you know what 2 of the wells are going to look like? Well, these organism, remember we played it on mannitol salt agar. So that means if it turns yellow, it can utilize mannitol. If it stays pink, it just doesn't utilize mannitol. And then also, because there's high salt in the media, we can tell whether this organism likes salt or whether it can tolerate salt. So I want you guys to take a look at the wells and try to find the mannitol. So I'll give you a little hint. Okay. And then I'll show you where the salt is. Up there. So you guys can go back and take a look at the wells and see whether it makes sense. And remember, my plate, my mannitol salt agar plate that had the microbe from my nose, did not turn the media yellow. It stayed pink. Okay. Okay, so I'm back and I'm ready to read the plate. I just need to put in whether it's turned purple or is borderline or is negative. And I'm going to do that on the computer. This is the plate I'm reading, which is the colony from my nose. And I'm going to put it into the computer and I'm going to indicate whether all of those wells are positive or negative or borderline based on the purple color. And I have to do this manually because that's all we have. Anyway, so I'm going to be back. I'll show you after I do that. Okay, so here's my plate that I filled out on the computer. And I put all the purple ones, the really obvious ones, as purple. And then when I was looking more closely at the plate, I found some that were really super faint. So I put those in as borderline. They probably won't even show up on this video. And unfortunately, as you recall, when I started this, I had fewer cells than I ideally would have liked. And the timing is just such that I can't leave the plate in any longer. So I've got what I've got. So remember that A1 is the negative control and A10 is the positive control. Those are very clearly negative and clearly positive. And as you can see, there's the plate again, just as a reminder, so you can see the really positive ones showing up right away. So let's see what it gives us. So we go to OK. And we don't want that. So in the green bar, you can see it says no ID yet, continue incubation. And unfortunately, like I said, I just am not able to do that. But I can see that it says that it's likely going to be Brevibacterium linens, which I'm not familiar with that organism, but because it has a 23%, the SIM value there is 23%. So the other ones are kind of farther behind. So it's more likely going to be the top, the first choice. And if we just incubated it longer, it probably would come up with that ID. So I'm not familiar with this, but I'm going to go and look it up and see what it says.